you said, Waylon Hastings and Christian Behrens. Thank you so much for coming.
Absolutely. Thanks for hanging out towards the end for all of us. My name is Christian Behrens. I'm a nutrition and exercise physiologist, and I'm also the Associate Director of U.S. Medical Affairs for the Consumer Health Division of Bayer, or Bayer, depending on where you live. Whenever I made the transition from academia to industry, one thing that was really important to me was being able to maintain my integrity as a scientist, being able to drive strategic medical decisions in the direction of ingredients and technologies that are supported by rigorous and reproducible science. I can pretty confidently say I found that at Bayer, where myself and other employees show up to work each day with our missions of health for all, hunger for none, and science that matters at front of mind. In the spirit of those missions, we regularly collaborate with external academic and industry partners to add credibility and confidence to our innovation and precision health efforts. Our collaboration with Hurdle in the longevity and healthy aging space is no exception there. In a minute, you'll hear from Dr. Hastings, who has collaborated with us as an outside academic expert in that field, alongside our partners at Hurdle to develop a novel saliva-based inflammaging biomarker. In addition to the biomarker, as a part of our healthy aging ecosystem, we've developed a cellular health dietary supplement and a interactive personalized wellness app, all to help consumers make informed and evidence-based decisions along their healthy aging journeys. This is kind of a key critical piece of our strategy for precision health, empowering consumers with education, diagnostics, and where appropriate, prevention. So with that, I'll kick it over to Dr. Hastings, who will highlight the development and validation of the biomarker, which is now commercially available, but I do not have a discount code for.
I'm glad you mentioned the commercial part because I was going to make sure and squeeze that in there. As Christian said, we've heard a lot through the last 2 days. Industry representatives, academic representatives. What I'm going to do here is try to both emphasize the actual science, this project, but really try to emphasize this as an example of how all of these different partnerships came together in different groups to develop this project. So here we go. So I don't need to belabor this point. We all know inflammation is heavily associated with chronic disease and aging. We can look at different mechanistic influences of inflammatory biomarkers on pathways implicated in aging. We can look at it with our favorite functional outcomes and health span characteristics. And we can also look at associations of these inflammatory biomarkers with other biological aging metrics. And again, not needing to belabor this point, all I did was choose one of my favorite images from the last couple years that represents this process. So here, I would consider these top rows, these to be our sort of canonical inflammatory biomarkers. Now, in recent years, we have taken a greater omic-based approach. So this is work out of George Murphy and Paolo Sebastiani's lab. And this is developing a approach to differentiate between centenarians, younger and older individuals, using differences in immune cell proportions. Earlier today, we heard about the IAGE measure, which was developed by David Furman. And so these are our sort of newest generation of getting an algorithmic approach to quantify inflammation. Now, when looking at these different measures and thinking about how we want to translate this both to the clinic and to the commercial sphere, we need to come up with some considerations for how we're going to evaluate these. So really important ones in this sense is stability. Is this measurement robust to noise from short-term fluctuations? We want it to be measuring chronic systemic inflammation across the lifespan. And is it precise? High test retest reliability. Is it stable? Is it something, or scalability, apologies. Is it something we can measure using widely assayed technologies so that it is easily implemented across locations, across laboratories? And is it easily computable? So both the practical logistic side of conducting the biological measurements, but also what is the computation that goes into generating the final output that you would use? And then, is it translatable? Is it measurable within patients in a non-invasive way? And is the biosample itself subject to minimal degradation at room temperature? Ideally, if we're thinking about a commercial sense, this would be collectible in an at-home kit. So considering all of these different parts requires very diverse expertise. So for this project, this is our team. We had representatives at Hurdle who were leading the R&D and the project development. We had teams from the academic side, including myself with my background in biological measurement reliability and the short-term translation of biological information across tissues. We had Dr. Janet Lord, who is an expert in running these clinical studies and helped with the reliability cohort data collection. We have experts and statisticians like Dr. Daniel McCartney with the Generation Scotland, which we heard from Ricardo's talk yesterday, who is the computational biologist doing some of our secondary validation. And then we have Christian, who is representing our Bayer team, who's really at the heart of pushing this all forward. So what is our motivation here? Well, first we have these classic inflammatory biomarkers measured in blood. They have lower correlation with age, they're more sensitive to confounders, they have decreased reproducibility because different assays and techniques are used to measure them, and they're lower accessibility and scalability because they're measured in blood. Now, we can try to make some progress along this front by developing DNAm epi scores that are predictive of these blood biomarkers. So this would have higher correlation with age, it's less sensitive to confounders because it's using these epigenetic processes, enhanced reproducibility because it's being measured on a standard array that can be reproduced between labs, however, still measured in blood, which decreases our accessibility and scalability. But finally, what we can try to do is to translate this knowledge from blood epi scores to saliva epi scores, which is what our motivation is with this project. And we're sort of fulfilling all of those different gaps. Now, here is the framework. So this is going to be the next 5 slides. That's just going to reveal different parts. So first thing we're going to do is to identify all of our data sets. In this sense, we have our training data set where we're developing the biomarker. We have the reliability data set that we're using to inform what different epi scores are fed into the biomarker. And then we have our 2 test data sets. So we generate our inflammatory epi scores in all of these data sets. And then what we wanted to do was filter for reproducibility both within tissues and across tissues. So I'm going to not belabor this too much, but we have this custom platform called the Hurdle Array. It's a custom DNA methylation array. It's only about 30,000 sites that's been pre-designed to be enhanced for these inflammatory probes, but also geroscience relevant sites. So different colors here represent different tissues, and the gray represents different platforms. What we're doing is establishing ICC thresholds at the level of the epi score for we want these information and these measurements to both be translatable across platforms. So this is the EPIC versus Hurdle within tissue, but also be translatable across tissue. So developing different ICC thresholds for this, we narrow down the actual epi scores that we would use for subsequent training. After that, we're going to select our features based upon biologically relevant criteria. So this is where Steve Horvath had in his first talk. The third sort of class of epigenetic markers we're seeing is they are developed by biological inference. They are theory informed. So in this sense, we have epi scores that are all tied to predict these different inflammatory biomarkers. And what I want to point out here is it's not just because they're correlated with chronological age that they're kept in the model. In fact, the top epi score, it failed our reliability metrics. So we removed it from the model regardless of how it might associate with chronological age, or it might have the strongest correlation with chronological age. So letting theory drive the decision, not a high or a low mean absolute error. Then we are training model on the selected outcome. So after we have refined our input data set, we are training then on chronological age. Why are we still training on chronological age? Because customers like a very simple number that they can interpret. So if you're going to give them their InflammAge, they want a number that they can understand. So here, this is in our training data set. We can get an R of 0.934, very highly correlated with chronological age, and a pretty low mean absolute error. And also what I want to point out is that if we look at the ICC of the InflammAge measure itself, we can see that when we do this ICC filtering at the level of reproducibility across platforms and across tissues, we have improved ICC of the final measure if we hadn't conducted those steps. And also more narrow 95% confidence intervals. All right, so we've trained it. Now we are testing it. And so this testing in independent data sets, we have two different data sets that we're testing this in. First is our paired blood saliva data set. So what we're doing is we have saliva samples. We've taken DNA methylation in those saliva samples. We have at the same time point from the same individuals blood samples, and we are doing blood inflammatory biomarker measurements on that. What we can see here is that we can reproduce the same correlation with chronological age in the first test data set. And what we can also see is that our InflammAge residual, so the adjustment for chronological age, is correlated with the number of biomarkers outside of the reference range. So these are ones that are outside of what would be clinically normal, whether high for inflammatory biomarkers or low for anti-inflammatory cytokines. Now, if we look at another cross-tissue clock, the skin blood clock, it does not exhibit this same degree of correlation. It doesn't have this level of prediction. Next, these are analyses from Generation Scotland. So these are translating the saliva-trained InflammAge measure and then using it on blood DNA methylation data. What I want to point out here is that we see changes, it is correlated with changes in immune cell distributions that are indicative of immunosenescence. So we have increased monocytes, increased neutrophils, increased CD8 effector cells. We have decreased B cells, decreased CD4 T cells, decreased naive CD8 cells, as well as global CD8 cells. Another thing I want to note is that this residual, although it produces this patterns in blood, is not correlated with the DNA methylation, the cell decomposition in saliva. But it is tracking into that blood. So it's able to cross those, the robustness of differences in immune cell composition that we see in saliva samples. Now, a lot on this plot. We have different disease outcomes that we're looking at. The thing you want to point or focus your eyes on is the dotted boxes point out the best predictor in that domain or for those different outcomes. What we can see is that we have InflammAge is able to outperform different measures in these different domains, and it's significantly associated. So the error bars don't cross zero when the boxes are around them. And we can see that we are able to exhibit robust associations with inflammatory disease outcomes. We can also see that we can have significant prediction of mortality across up to 16 years of follow-up. And that the effect sizes are outperforming, again, the skin blood XL, and is generally equivalent or equal to CRP, except in the case of the 2-year short-term follow-up. So in summary, this project is just meant to be, in addition to be a really cool new inflammatory biomarker, but also serve an example of how we can come together as academics, research, and industry, and pharma to create these new novel approaches to biological age assessment while we're taking into account all of these different considerations. So academic rigor and reproducibility, practical considerations for large-scale implementation on the industry side, and targeted to geroscience and longevity for the pharmaceutical interests. This InflammAge biomarker is targeted towards systemic chronic inflammation, and it has translational potential based upon the being both readily accessible tissue and having robust associations with hypothesis-driven outcomes. And then finally, future steps is, as was previously mentioned, getting this integrated into the Biolearn platform so academics can freely use this measure in their studies while protecting the intellectual property of Hurdle. And if you would like to scan this, our preprint is available now, and hopefully in the next month, our publication will be out in Aging Cell under re-review right now. And with that, I think there's a minute left. All right.
Thank you, Waylon.
Yeah, thank you.
Thank you, Christian. It's really cool. You should put it on Biolearn.
Okay. Yeah.